Is that what it is? What did it produce?
So we're gonna still have a class this week. Today we're gonna talk about immunology, and then we're also gonna go ahead and talk about the virus. If we have a time, then Thursday basically we finish the virus, then we're gonna do the review. And I will go ahead and put the key on the e-campus this afternoon. So some of you are taking the early exam on Thursday. On Friday, you certainly can start to work on it. Okay, so I'm going to do that. But we're still going to talk a lot today. We will talk about called immunology. So what is that? You more or less heard about that. This is coming from a word called immuneness. So what that means? That means free of burden. Now, what did this burden comes from? This means foreigner. So for your body, any of those foreigner coming, our body will react it, and we are trying to take them away. So that's a good thing for prevent, or you can say curing from pathogen, from our standpoint. But they also have a bad thing. If you are organ transplant person, then the first couple of days or the first couple of months and the 1 or 3 years, you're going through a tough process because the transplanted organ will treat it as a foreigner, and the immune system will repair it. So that's a challenge. So challenge for transplanted patient. So there's a good thing and there's a bad thing. And also, lots of you know, lots of your friends, more or less, have some of the reaction to peanut butter, to fish, to seafoods. What do we call? We call it allergy or allergic. This is also is an immune response, but we say it's an over response of the immune. So allergic, that's another bad thing. Okay, so that's what it means by the immune. Now, in our body, if you want to talk about that, I'll skip a couple of slides and then I come back to talk. We basically will have is two different immune systems. Is non-specific. And we have a specific immune system. Now, non-specific, we already know, you're already feeling about it. So, for example, if we are sweating, you feeling about there is oil comes out from your skin. That is type of the non-specific immune to prevent the pathogen. Colonization. And you see there is a salt comes. If you run the half marathon and you see the sweater completely wet, and after a couple of hours you will see the salt is extracted. So obviously the salt is on your skin surface. That is a prevention of the pathogen, because not all the bacteria are halophiles. And let's say you do a coffee. That's an action also to take out of the bacteria. Even we go to restroom, we do the flushing. That is also taking out of the harmful material from your body, which is the formation of liquid, which is called urine sample. And we also have a very established bacterial community in our gut area. That's also be a natural immune system. And lots of the research has been showing that will be the center of your body. And we also see a big chunk of white blood cell related to macrophage That is also our non-specific immune, which means everything comes, they are going to start to work, start to attacking the foreigner to get rid of it. So let's spending less time here. I'll go back to talk about slides. Because in our exam, we're going to focus on the specific immune. So let's talk about this first. We go back to talk about these slides. So let's talk about the specific immune. So let's talk about acquired development of the human immune system. So first of all, you need to know, we had two different lines of the immune. They all coming from stem cell. The stem cell, if they go to bone marrow, and then they are going to two different major immune organs, which is spleen and lymph nodes, and then they will generate so-called B cells. This we call humoral immune. Humoral immune will generate antibody. That is the key. Now, where the B comes from, in the old time, about like 150 years ago, there is a scientist called Bursa Fabricus. It's a Spanish scientist. So one day he did the research, which is get the tissue cell culture from the butt area of the chicken. And then he isolated the B cell and purified the antibody. So in order to memorize him, we took this B out, we call it B cells. The key thing is it will generate an antibody. Now, the stem cell, if they go the other direction, which is go to the thymus gland, and also later on transfer to spleen and lymph nodes, this will generate T cell. And T cell is different between the B cell. They do not generate antibody, so no antibody. What we call this one? We call it T cell mediated immune. Now people think you are smart, so they take this T out. They become cell mediated immune. That is two major immune system, two major specific immune in our body. So we call B cell line and T cell line. So this is a big picture. Now, if we are specific to talk about a special antigen, because once you have antibodies, they must have something to stimulate, to generate, that is called antigen. Then it's could be talk about a little more detail. So this means we have an antigen. Okay, we'll talk about the antigen. Let's say we got a bacteria, or we get a virus. So they go to the body. And when they go to the body, at the very beginning, they will going through to find a cell. It looks like this. Looks like a leafy. So what is this called? than the Retic cell. And then the Retic cell has lots of the very tiny complex cell, like this, or like that. Lots of. What are they? These are all called MHC, major histocompatibility complex. Histocompatibility complex, called MHC. This MHC is to recognize the foreign. They transfer to dendritic cell. So dendritic cell, we also call it antigen presenting cell. We have another name for that. Now, based on the size of this antigen, we have two lines for that. If the antigen is relatively small, we call it a small soluble antigen. Then they will go ahead to find a B cell. So how do they find the B cell? We talk about the antibody. On the B cell surface, they have lots of stuff like this. It goes here. They could attach to it. And usually they go here. We'll talk about antibody structure very soon. Okay? They go here. Now, first of all, this B cell is not only one. They will go in through multiplication, and we call it cloning. So they will generate lots of the small B cell. Lots of small B cell, like that. Now these small B cells could also be deassociated. They could be only like that. This is what we call a plasma cell. And then they could generate something like this. That's antibody. And then they also could generate is a whole compact structure, which means they have everything after multi-cloning. And this is B cell. So what is this one called? Memory cell. So once the B cell come out, they're going to go through multiplication, the cloning, they have antibody. They have a plasma, isolated plasma cell ready for another antigen coming, antibody to attach to it, and they have a memory cell. This memory cell could release the antibody all the time because they already like a bank storage in the cell membrane system or cell wall system specifically for the bacteria. So this is also the line we call the B cell. But this is more specific in an example when an antibody is coming. But how about if you have a big one? Okay, if I have a large one coming, this we call it a large complex antigen. Once we have a large complex antigen coming, Instead of they activate the B cell line, they will activate the T cell line. And they will be created lots of their daughters. So for example, we have a natural T cell. We have a T killer cell. We have a T helper cell. And we also have a T memory cell. So here we have one thing we want to be very clear and emphasize. The major difference between T cell and B cell is the antibody generated. It's not the memorization. T cell also have a memorization, but the memorization time is very short. Okay, and but T cell does not generate antibody. So that's how the acquired immune system developed. And the human immune system, in a very brief picture showing you how they developed. I have a very good picture. I find it in an old textbook. And I already take a picture and I will be uploaded onto the eCampus. Then you guys can refer to what we just talked about. So we have an antibody. We have an antigen. They will have an activated immune system created. Now, what are the major formats for the immune response system? We know there is artificial acquired immune. There is a natural acquired immune. So basically, there are 4 different categories. The first one is called natural acquired active immune. So what is that? How will we understand the natural acquired active immune? It's very simple. Is recovered from pathogen. Or we say pathogen caused disease. So for example, you are recovered from the pneumonia. Typically we come from streptococcus pneumonia. Then, after a certain period of time, if we isolated the blood cell, we will see it's rich in anti-streptococcus pneumonia. That is called natural acquired active immune. Then you have a second category, which is we call natural acquired passive immune. What's the example? Natural acquired passive immune. So think about this way. The newborn baby usually or theoretically in their first 3 or 6 months, they're usually free of any type of the disease, especially infectious disease, because they will get the IgG from the mother. So the example is placenta to the newborn. So for example, the IgG, and this is called the natural acquired passive immune. Why we say it's a passive? Because it gives from the mother to the newborn. So since we have a natural acquired, we should have artificial. So we have then we go to artificial acquired active immune. Okay, what's that? Vaccination. If you get flu shots, that's artificial acquired active immune. because that's what you go to do. It's very active. You get the shots and they generate the antibody. So the same thing, artificial acquired passive immune. What's that? This is a very real example, but we don't usually happen in our life, but occasionally we will do. So remember, when we talk about the Clostridium botulism, if we have a Clostridium botulism, what do we have to do? We need to do antibiotics. But before that, we need to do antitoxin. And I say the antitoxin is anti-equine toxin shipping from the Atlanta airport. So anti-equine toxin, at this moment, if it's given shots to the Clostridium botulism patients, we call artificial acquired passive immune. The reason is you must get that, otherwise you could be died because of the Clostridium. So these are the four major categories. What we talk about is the immune system happening in our body. Now we want to talk about what is antigen. So I'm gonna remove this, and we're going to talk about the antigen, and also we're going to talk about the antibody, and then how the antigen and antibodies are going to react with each other. So I had a much better picture of here, so I'll just remove the whole thing. So let's first talk about the antigen. We are microbiologists. We already studied microbiology a whole semester. So there is something we want to talk is a whole bacterial cell. This is not an antigen. If we want to talk about the antigen, which means they are specific structure. So, what are they? I'll give you a very easy example. And you should know right now what are they. What is H? That's flagella. What is O? Modified sugar. Ceramic. And we say the side chain of polysaccharides of the cell wall. 157H7 is serological typing. So, what this tells you, when we talk about the antigen, that means a specific structure. For example, the modified sugar, which means a cell wall, you can say. And flagella is a structure. That's an antigen. Another thing, do you remember Streptococcus ammonium? And we say this one did not belong to any of the Rebecca Lance field group. Why? Because Streptococcus ammonium, there is no M protein in the cell membrane system. And this M protein, that's antigen. So this is a very good example, tells you a specific structure of antigen. Now, here is another picture which tells you. You have H antigen, O antigen, M antigen, plasma membrane antigen, ribosome, K antigen. K antigen is a lot. So in the older time, or you read the manuscripts of the research, so we talk about O157H7, but sometimes you will see K88, K89. That is specific means a capsule. So they do a serological typing of the capsule instead of the flagella and the modified sugar. So that's also verified what I just said. The antigen, which means the specific structure of the bacterial cell. It's not a whole cell. Okay, that's antigen. So what is antibody? So first of all, antibody is a protein. So what it looks like? Like this. Y shape. This angle 120 degree. You have this line is very long. Or just go like this. Very long. We call it H chain or H line. So why we say it's a heavy chain or heavy line? Because the molecular weight is higher. It's about 50 to 75 kDa delta, which is used to measurements of the amount of the protein. Now you have this line on the side. This is we call it a light chain. 25K delta. Delta. Okay, so that's a basic structure. Now how they connected with each other? Lots of the time you will see that SS there, SS there, SS here, SS here. Maybe a little bit here sometimes, not always, a little bit. So what is that? Disulfide chain. Okay, they connected with each other. Now, we talk about antigen, we talk about antibody. Antigen and antibody will be reacted and they connected with each other. How they connected? You're right here. So, what's the location called? This is called variable area. And that is attached to the antigen. Now how about the here? The tail is very long. So we call it an FC segment. What does FC means? Crystallized fragment. That's acronym F and C. Now what the FC segment for? The FC segment is for connecting something like that. This is called macrophage. So we talk about so many. So basically information. So remaining question will be how antigen and antibody reacted, how they interact with each other. We have another slide for that, which is right here. There are majorly 5 different interactions. Number 1, precipitation. And this is, you already know what it means precipitation in a tube. If you have an antigen and antibodies, they reacted with each other. By the way, usually they have to be specific. Okay, you have Streptococcus pneumoniae, then your antibody have to be Streptococcus pneumoniae. So that's called precipitation. So if you have a tube, they reacted with each other, you do a centrifuge, and most of the time, they will be at the bottom after centrifuge. The second example, we call it neutralization. Neutral. So what means neutralization? In other words, it's equals antitoxin. So let's say you have equine, anti-equine toxin. You have a Clostridium botulinum toxin. They could be reacted with each other to neutralize the botulism toxin. This is the antitoxin. We call it neutralization. The third thing, agglutination. Agglutination is another formation of the precipitation. Instead of they simply stick to the bottom of the tube, they're going to forming something like that. We call clumping. This could do a good job of testing bacteria. Remember your final lab exam. I have a question. I said I didn't review it. I'm testing you whether you know from the other classes, and we very briefly mentioned it. So the question is, from your throat culture, you isolated the Staphylococcus aureus. How you should do the confirmation? And remember, at the beginning of the exam, I said, I'll give you some hints. You can use the exclusion methods, which means it's not an A disc, it's not a P disc. So what it is? The answer obviously is D, is latex agglutination test, because using clumping to see the antibody and antigen has a big matrix on the testing paper, or we say testing kit, and we will know whether it's Staphylococcus aureus. And this one actually coming from the experimental animal, which is a rabbit. So we want to talk about that. We'll talk about real quick how they generate it. So that's called agglutination. Now, antibody and antigen, lots of the time, they reacted with each other. But for our body to really repel or against the foreigner, it's not only with the help of the antibody. They need other things to help. Therefore, we have another terminology right there on the top. You can see it's called opsonization. What means opsonization? Which means antibody and antigens are reactive. As I just mentioned, the structure here is functional. So you always remember a certain structure in certain microbiology or biology entities, they always have a function to that. So where is this Fc segments for? If your antibody and the antigens are fighting with each other but not going to solve the problem, or it's a tie, you have a tiebreaker. The tiebreaker is a macrophage. They're going to bring the macrophage, and the macrophage attached to Fc segments. And this reaction we call opsonization. So said very simple, this is bring macrophage to Fc segments of antibody and activate more antigen-antibody interaction. So that is called opsonization. Now, not only for that, lots of the time you think about the animal or human body as a whole entity, we have lots of the immunoactive chemicals in the blood cell. An example is called complex. So complex, this is a small group of immune active, you can say it's a protein. Therefore, we have C1, C2, all the way go to C28. once you antibody and antigen are fighting with each other, sometimes you bring complex. This is called complex fixing. And what they end up with is the bursting and the lysis of the bacterial cell. So you can see. They reacted with each other, and they bring the complex. And the complex gives them a vendor. Looks like it gives them a stage to let them fight it. And when they fight it, what happened? They cause the burst and the lysis of the bacterial cell. So here, what we give you the example is 5 major interaction between antibody and antigen, which is precipitation, neutralization, agglutination, opsonization, and the complex fixing or complex fixation, whatever you call it, that, which means it's bring the complement. Okay? Sorry, that should be called complement. I was misspoken here. This should be called complement. Fix. Not complex, it's called a complement fixer. So that's antibody and antigen, how they react. Okay. Now, we already talked about the vaccination. So we just want to be refresh what we already learned. Because we talk about this very beginning in our first lecture. What is a vaccination? Which means they stimulate an immune active system in your body. And lots of the time you can understand is you give a shots, you get a low dose of the pathogen. And the pathogen will not cause a problem of your own, will not have an illness. But they did activate the immune system of your body and stimulate the antibody-antigen reaction. And then they're going through multiplication and cloning, they generate a huge amount of the antibody. So that's the immune. Now, if you get shots from CVS or from Walgreens, let's say, or from your hospital, that's basically we call it a vaccination process. And we give the category already that is called artificial acquired active immune. Now, what type of the vaccines we have? And I listed here, give you some of the examples. So the first one, what we can do is kill the microbes, which means dead cell. What that means, the cell is dead, but the antigen is still could be existing. So let's say polio and the whooping cough, those are killed microbes. Now killed microbes, basically, they only activate is T cell line. And the memorization is relatively short. That's why the whooping cough, remember DTaP, we have to give a booster all the time because it's activates only the T cell line. The second one is attenuated vaccine. What means attenuated? Attenuated basically is a live cell, but a low dose. So, since it's used a live cell, the stimulation in your body is very strong, or it's stronger than use killed bacteria. Therefore, it usually activates the B cell line, so you only need one shots in your lifetime. A very good example is MMR. What is MMR? It's measles, mumps, and rubella. And every immigrant or foreign students go to study in the United States, you're required to have the shots of MMR. It's one shot, 3 jobs, and it's only one time because it's a attenuated vaccine, activate B cell line. The next one is purified antigen. What means purified antigen? So remember this is a bacteria. Bacteria have a very heavy capsule for lots of the pathogens. Let's say Streptococcus pneumonia. So people can get a capsule, and then they isolated them to create a vaccine. This is called a purified antigen. For example, meningitis, HIV, we call it a type B Haemophilus influenza vaccine. That is a purified antigen. And whooping cough, that also could be from a purified antigen, from Bordetella pertussis, if you still remember. The next one, toxoid. So when you see tetanus, diphtheria, remember we talk about DTaP? This is toxoid. What it means? Toxic-like chemical. Toxic-like chemical is manufactured using chemical biosynthesis or manufacturing process. So they generate, we call it a toxoid. Last one, hepatitis, usually genetically engineered. That's why it's a little bit expensive. Sometimes insurance is not really covered for the hepatitis vaccine. Now, when we have these slides, the textbook has not been updated. But if you look at a very new, recent textbook, they talk about the coronavirus vaccine, where they come from. I kind of forgot a little bit because I was really feeling tired. We heard about it all the time in the last couple of years. If I remember, it's coming from the S protein of RNA. So that's brand new, because nobody has ever been touched before, because traditionally the manufacturing of the vaccine is going through those 5 parts. But they isolated the S protein from the RNA. But if you look at the news, our new FDA regulation maybe could say that's not very safe to use. So there's a debate about that. But I just want to let you know it's a new creative thing to have a vaccine of the coronavirus. And this is we already did, is immunize the hosts. Well, I just want to let you know there's a debate about this one also. This could be gone a couple of years, maybe 2 years later when I teach this class, I no longer talk about that. If you look at the news these days. So basically, the newborn baby in the United States, after 2 months, they need to go to see the doctor, which is their PCP, we call it. And then you're going to have to schedule your immune shots. So you can see DTaP, you have to do 2 months, 4 months, 6 months, 15, 18 months, 4 to 6 years. And I did it, I remember it's 12 years, you have another shot. I just did it, I'm like 45, so I just did it like last year. So you have to do it after 20 years, 15 years, do another one. Haemophilus influenza, that's you have to do 2 months, 4 months, 6 months, and 12 to 18 months. So you see pneumococcal, you see poliovirus, all these, they have a very detailed schedule for let you to do that. So just want to let you know, we do have a very good immune system about that. So last slide here. We talk about agglutination. We talk about the counting. And we also have a question to test your knowledge about how we testing Staphylococcus aureus. So how we really do it? There is a commercial kit which is called latex agglutination tester. Latex agglutination tester kit. What it looks like on the top. So the company called Rimmel, and they give you a tester kit, and it basically is the tester paper like that, a couple of holes, and you need to add 1 drop of the latex agglutination reagent. This is already consisting the antibody. Usually we call it anti-rabbit Staphylococcus staph antibody. Okay, we'll just say like this. People think you're smart, so took this on. And you add a reagent, then you use loop to add a bacteria, and you mix it. And then you shake the tested paper, roughly about 120 RPM, do a like circle. If you can see somehow, like showing in the picture right here, you see those clumping, which means the tiny dots, and they showed up, they're called clumping. That is a matrix of antibody-antigen reaction. That means the pathogen you put it there is matched the kit, so it's Staphylococcus aureus. So this is already being commercialized. You can buy it, and although it's very expensive. It's about like $100 or $200 for 50 samples. So basically $2 a sample. But in the history, this is not very easy to do. This comes with a rabbit at the very beginning. So I just want to let you know how we did that. Long time ago, I worked with one of our PhD students in China. They tried to do that. So let's say I have a rabbit. Okay, just a brief picture. Where we got the rabbit? Of course, the farm. But the farm usually it's a medical experimental farm. They supply the rabbit is relatively uniform to do the clinical research. How they do it? They give the shots here in the butt every day, or sometimes in the muscle. They give Staphylococcus aureus. That Staphylococcus aureus obviously is not a live cell. If the live cell goes there, the rabbit will be dead. Those are very vulnerable and weak animals. So what we do heat injured cell. Usually those are 70 degrees Celsius heated already. So what this means, the bacteria is not really survived, but the antigen is there. So we give a shot there. Then we need to get the blood. Where we get the blood? We dig a hole in the ear. We get the blood. So, have a tube to get the blood. And then your blood goes here. So you know about the blood, what's the components of a blood cell? You have RBC, is that right? Red blood cell. Then you have WBC, white blood cell. So by the way, RBC, what do we call it? S, right? What is WBC we call it? White blood cell. What do we have? Remember we have a basophil, neutrophil, we have a eosinophil. Is that right? Then neutrophil related to your antibacterial. Basophil basically is an anti-protozoan. Eosinophil is for anti-inflammation. So let's say you have Streptococcus pneumoniae caused the disease and you go to see a doctor. If they really run the whole blood sample of the patient, they will see you have a much higher than normal basophil because that is for anti-Streptococcus pneumoniae. Now what else we have in the blood cell? Platelet, is that right? Because this is why the cell can clotting. So we put it there. In the room temperature, about like a couple of hours, it will separate. What are they separated? On the top, it's a light liquid. We call it a saline. And this salient ideally should be rich antibody. And how we tested it? We using 96 well cell sample, cell plate. And we do a 20-fold serial dilution, like that. Very similar to like we did the serial dilution. So we add antibody, we add antigen, we add Staphylococcus aureus, we add our isolated the blood salient sample. We try to see is there agglutination happens. And we need to find the first tube, which is clear. That means that tube is rich anti-Staphylococcus aureus antibody. Can you see the dilution factor there? It's a reciprocal number of the dilution factor, and that's called a titer. So what we want the titer to be smaller the better. Because much smaller, the titer we get it, which means there are huge amount of the antibody there. That's why we need to do a huge amount of the dilution. But most of the time, we get very high concentration, very low concentration. So our titer lots of the time is small. It's like 20 or 40 or even 50. But lots of the time what happens is the rabbit is dead. Even you give a couple of shots. Now, there's another thing we do. The rabbit is dead. We give the shots. So we need to give some carrots. So we give some carrots. Let the rabbits to eat. There are thousands of carrots we are shipping from the farmers' markets to let the rabbit eat it. But unfortunately, lots of the rabbits has already been dead before we can get the salient sample. So what we do, we get heat-injured Staphylococcus aureus. We try to lower the dose, lower the concentration. Now there is also some of the rabbits are really strong. No matter what you do, they never die. And then but they don't generate the high amount of the antibody. So what we do, we keep giving the heat-injured Staphylococcus aureus. We call it booster. We try to give them more toxic to let them generate antibody. So often the times, it's need about at least 2 to 3 years they could generate the titer number is 160 to 320. That's a very commercial, that's a very economic benefit material. So back in 20 years ago, 1 ml of anti-rabbit Staphylococcus aureus, it's about $25,000. That is enough to pay you WVU tuition for 2 years. By the time maybe it's 4 years in the in-state student. Later on, people developed lots of the methods called the epidizer process, generate lots of the antibody. And later on developed the latex agglutination testing kit. That's why today we could using these methods to test the bacteria. But in the older time, people need to spend a huge amount of the work to do, and at least 100,000 of rabbits will be sacrificed. And this whole thing could be a PhD dissertation. There's only one paper, which is you isolated. successfully isolated a certain pathogen from the rabbit salient solution. So that is what our last slides for. That is why I want to have you testing in the lab exam for that question, because there's a big story behind it, how we get the latex agglutination testing kit. So we haven't finished here, but we have one thing we haven't mentioned in very much detail. We want to go back to talk about here. So we say, we spend lots of time talking about the immunity. We talk about the humoral cell-mediated immune. We didn't mention too much about our host resistant, which is our natural non-specific immune. So here gives you some of the examples. I mentioned the skin, coughing, flushing, but don't forget our mucous cilia will also the natural defense system. Low pH, when you see low pH, what this tells you? It's a stomach acid. Stomach acid is very low, the pH is 1 to 2. And that's why we say HP bacteria, they could be neutralized, the low pH, because they have a ureas. They can break down urea, become ammonia. That's what we talk in the lab. The lysosome. The lysosome, we have also a picture for that. It will create a process called phagocytosis. So what happens is, if you have a bacteria go into the body, the first thing what they do is macrophage. A macrophage is one component of white blood cell, or we can say one member of the white blood cell defense system. So that when they come in, they will eat it. That's what we call it engulf. And the lysosome will attract into it and then generate a phagosome. And the phagosome will lysis the bacterial cell. Now how they do? They'll break down 1,4 glycosidic bonds. And basically that is the glucose. So that's how they do. And also they break down the N-acetylglucosamine and N-acetylmuramic acids. So we already talked about that, so we just be very brief how the lysosome works. We also have lots of other things. Talk about normal microbial flora and the microenvironments. This is our normal human microbiota. And you can see how huge it is. Lots of them we already talked. So nasal area, Haemophilus influenza, Streptococcus pneumoniae, and Corynebacteria go to the mouth. Streptococcus, we talk about Veridium Streptococcus in our exam 3. Skin surface, Staphylococcus, Micrococcus, Ledius. It's a tetrade coccale bacillus. The vagina tract have a huge amount of the lactobacillus. So that's why the pH is lower and also the preventing system for the pathogen. But there's an exception if it's very dry, which is using highly absorbent tampon, will cause a problem, which is Staphylococcus aureus. So urethral. So you go to the throat area, you see so many normal bacteria there. We mentioned lots of them, Neisseria, Haemophilus, Corynebacteria, Staphylococcus, Streptococcus. But if you look at the stomach, very few bacteria. Look at the HP, Helicobacter pylori, the pathogen related to the stomach cancer. Small intestinal, we have some bacteria there, not too many. When you look at the large intestinal, that's huge. So, recently, if you even working at the WVU Health Science Center, there are some faculty will using 16S RNA biotechnology to do the analysis of the normal bacteria community in the intestinal area or in the guts area. That is the ongoing, very hot topic, because people are thinking, in addition to our brain, our heart, our intestinal the microbiology community is the third, or even some people think the most important center of your human body. So just want to let you know, this picture, there, we can talk this a whole day. There's a lot of story behind it. So, white blood cell, we mentioned. That is a blood cell. So keep in mind, we talk about the B cell, T cell, that is also part of the blood cell, coming from lymphoid. But most of the time when we talk about the blood cell, we talk about this red blood cell, S right side. We talk about white blood cell, basically is granulocytes, macrophage, and platelets. So we talk all the time is on the left, 6 members, the B cell and the T cell, we did not notice that. But today we're spending lots of time to talk about it. This is the system. You can see the bone marrow system, the lymph nodes, and the thymus gland, that's all the spleen, that's a system we exist in the immune active organs. We already talked about that already. Here we have a couple of terminologies. You should be easy to know what is called the immune system, what is called immunity, what is immunology. Okay, so basically we talk about it already. We have another slide right here. It's called a non-specific surface defense system. On the very top, you see the conjunctiva. It's an action of the tear. So there's an epidemiology and social studies that has been showing. So part of the reasons the women have a longer life than the man, because they are sometimes always crying. Because when you're crying, you generate the lysozyme. And the lysozyme is a very natural defense system in our body, which could be actively working in the first stage for the non-specific immune defense. So that's why we have this picture here, talk about the conjunctiva, which is have the lysozyme. Then all the others we already being mentioned. So that's another good picture to understanding the non-specific surface defense system. Okay. Last thing I want to mention. When you have a microorganism into your body, you should not feel the nervous. It's not really means you have a disease. There is a different stage, and you can see. It could be just a repair from the body, which is transit. But most of the time will cause infection. Now the infection will result different results. It could be become a normal bacteria. It could be inapparent infection, which means there's no symptoms. Or it could be a carrier, or finally be eliminated. But most of the time we think about it's a bad thing, because 90% it will be end up with infectious disease. That's a reason. Okay? So that's all we have for this section. What is the key point for this section is I drawed earlier for the development of the active immune system. If you want an extra point, the structure of the antibody you should know. You should know how to draw it. With the interaction between antibody and antigen, I do want you to know what is called opsonization, because that's very special. Just bring the macrophage to the FC segments, and the macrophage, that is part of the components of the white blood cell. So that is a very special terminology you should know. And you also should be very clear about these slides. It's an outline to talk about the non-specific surface defense system. Okay, so that's all I have. And Thursday we will very quick half an hour talk about the virus, then we're going to do the review.